Right, so in this video, I'm going to be going over synaptic transmission between two neurons. So here we have a terminal end right here of the first neuron on the presynaptic neuron, and the dendrite right over here of the second neuron or the postsynaptic neuron. And in the middle, right here, we have the synapse and the synaptic cleft, which is the communication point between neurons. So let's just say an action potential reaches right here, the first neuron, and triggers a voltage gated. It'll keep going right here, and these sodium channels will keep pushing it forward until it reaches these things right here. These are actually voltage gated calcium channels that will open up due to the action potential. And it'll push calcium right inside here. The presynaptic neuron also has these things called vesicles right here that have neurotransmitters inside them, these little green things. And what the calcium does is it floods in and bonds to these proteins right here that are holding these vesicles in place next to the exon terminal. These docking proteins are called snare proteins. The calcium changes the conformation of the proteins just enough so that the proteins bring these vesicles closer down, straight down here, to the presynaptic membrane. The snare proteins will then pull the presynaptic membrane and the vesicle apart so that the two merge. Basically, you'll get an opening like this, and it'll merge, and then this will disappear. This will allow the neurotransmitters right here to be dumped into the synaptic cleft. They will bind on the surfaces of the postsynaptic neuron. They will bind to the receptors on the postsynaptic neuron that are specific for those neurotransmitters. And they'll work like a lock and a key right on top. So they're going to bind right there. That bonding will then trigger ion channels right here to open, allowing ions such as sodium to flow into the postsynaptic neuron. This is an excitatory response, but there's also an inhibitory response as well. The inhibitory response would come from the neurotransmitter triggering a potassium channel. So let's say this is potassium channel. That would bring an efflux to flow potassium out of the postsynaptic neuron, causing the neuron to be less positive. The neuron becoming more negative will make it even harder for an action potential to start down here and go down. Hence the inhibitory response. Now, if the action potentials are being fired too fast or too frequent, then the rate of neurotransmitters release may exceed. So these action potentials are going way too far. It may exceed the amount of neurotransmitters coming down here. So there'd be a ton out here. So to get rid of the excess number of neurotransmitters, the first thing that may be is diffusion of the neurotransmitters out of the synapse. So they'll diffuse by simple diffusion straight out here into the open to get rid of some. Other forms that are an active form of removal, instead of passive diffusion, are the enzymes that break down the neurotransmitters inside here to be recycled. The next active form is through a special pump on the presynaptic right here, on the presynaptic membrane of the neuron, and that pump the neurotransmitters straight back in to be recycled. Another way to be recycled neurotransmitters is through the astrocytes down here. So these astrocytes are part of the central nervous system. These astrocytes have a special pump on them, actually, that takes the neurotransmitters and pumps them out into the astrocyte to be recycled again and either brought back into the presynaptic neuron, could be used again, or for other things. Lastly, we're going to talk about how the neuron returns to its resting potential after it fires an action potential. So the neuron returns back to its resting potential via repolarization. This is done through the inactivation of the sodium channels. So these stop working. So sodium can't get pumped in. That'll stop the inward rush of the positive sodium ions. At the same time, potassium channels are opening. So K channels over here are opening up, and K is actually leaving out. The loss of the potassium promotes the repolarization, which is known as the falling phase of the action potential. The neuron returns to resting potential, but actually overshoots it and becomes hyperpolarized. During this phase is known as the refractory period, and it is very difficult to cause the neuron to fire again. And so basically the neuron can reset itself. Eventually these potassium channels will close, And the membrane will return back to its resting membrane potential of negative 70 millivolts to be activated again.